Good afternoon, everybody. My name is John Bischof. I'm the director of the Institute for Engineering and Medicine, and I'd like to welcome all of you here to our annual Otto Schmitt Distinguished Lectureship. So I just have a few slides to show you just to get you grounded in what the Otto Schmitt Lectureship is and who Otto Schmitt is. And I can see by this, the way that this classroom is set up, that I'm going to have difficulty making eye contact. But anyways, this will be great. This will be an experiment. Yeah, so I will go through the slides to tell you a little bit about the Otto Schmitt. Then I will introduce our guest today, and then I want to just point out that we will be hearing from Dr. Edelman for roughly about an hour. But then we do have time for networking. So if you haven't had a chance to meet him yet, you will have a chance afterwards if you'd like. And so I'm sure he would encourage you to come up after if you have questions. There will also be time for question and answer. Again, welcome. So this is our one distinguished lectureship that we have every year in the Institute for Engineering and Medicine. And it's in honor of Otto Schmitt. And so he actually was a longtime faculty here at the University of Minnesota, and I'll tell you a little bit more about him. And also, if you don't know about IEM, we're really here turning science and engineering into medical practice. That's our mission. So we're fostering discussion around the grand challenges for engineering and medicine, and this is based on bringing in some of the very best national leaders in research, industry, and government in biomedical engineering to give this talk every year. And just to tell you a little bit about Otto Schmitt, he was a longtime university professor here in biophysics, bioengineering, and electrical engineering, and he made significant contributions across a wide range of fields, including submarine detection, electrocardiography, and quality of life measures. He's also credited with founding a large number of societies, professional societies, including the Biomedical Engineering Society and the Biophysical Society. So today we're pleased to welcome Dr. Elazer Edelman. And I have a few prepared remarks. And Andrew, this might be the time to flip out the... Yep. Okay. So it's my great pleasure to introduce Dr. Elazer Edelman to give this year's Otto Schmitt lecture. Dr. Edelman must be one of the crown jewels of MIT. He has 4 degrees from the Institute, 2 bachelors. One is in electrical engineering and computer science, and 1 in applied biology, 1 master's in bioelectrical engineering, and a PhD in medical engineering and medical physics. Along the way, he also got an MD from Harvard. Dr. Edelman is currently the Edward J. Poitras Professor in Medical Engineering and Science at MIT, Professor of Medicine at Harvard Medical School, and Senior Attending Physician in the Coronary Care Unit at the Brigham and Women's Hospital in Boston. Dr. Edelman directs MIT's Institute for Medical Engineering and Science and its Clinical Research Center, as well as the Harvard-MIT Biomedical Engineering Center, and he is the Associate Director of the Boston Biomedical Innovation Center. He also has adjunct or visiting appointments at Tufts University, the College of Engineering and Informatics in Galway, Ireland, and the Norwegian University of Science and Technology. Dr. Edelman and his laboratory have pioneered basic findings in vascular biology and the development and assessment of biotechnology. More specifically, his research is focused in 5 areas: polymer-based drug delivery, vascular and endothelial biology, tissue and biomaterials engineering, cardiovascular reactivity to implanted devices, and mechanical assist devices. He's published over 390 peer-reviewed articles and has over 80 actual or pending patents. He has advised or mentored well over 250 postdocs, medical students, graduate students, and undergraduates. I could go on, but of course we all came here to hear him. So please join me in welcoming Dr. Elazer Edelman.
Thank you very much, Professor Bischof. Kind of introduction only my mother would have thought was inadequate. But, I really am honored to be here for a whole variety of reasons. One, because this university has always been the house on the hill. You know, I live in in a city with 51 colleges and universities. So if one of them doesn't do something, there are 50 others wanting to. But this is the place that has to do for everything in this entire region. And the second reason is many of the people whose science I have admired, I finally have met in person. But the third is some of the people who accompanied me on my journey are here today. Will Durfee, who I met as an undergraduate, Ron, who was a lab mate. We're single-digit, first-hand members of Bob Langer's graduate cohort, and then, of course, Rick Coombs. But what I'm going to talk about today, and here are my affiliations, and I refuse to see them as conflicts because fundamentally everything that I think has been accomplished in the last 150 years has been a handshake between academia and industry. And I'm very proud of these. What I'm going to do today is talk about how the spirit of Otto Schmitt is inherent in everything we did, whether we knew it or not. And I'm going to use as a sounding board the fact that cardiovascular disease in our lifetime has undergone this profound changes. Still the #1 cause of death and disease around the world, wherever you live. But when we started, and the we is many of these people in the room, you either moved to Arizona or California to get out of the humidity of the northeast rather, or you had bypass surgery. And all what happened was when this man, Andreas Grunzig, in 1979, thought of using the balloon, and that's the original balloon that was used, and that's the original angiogram highlighted here in yellow is the stenosis in, does this show up? Well, in yellow is a stenosis of the major artery, and then that balloon opened that artery. What Grunzig said or thought would be a trip to the operating room, but it stayed open for 10 years. The problem was at the time that these things would clot and these things would fail. And they would clot and fail more often than they would succeed. That they succeeded was a miracle. But along the way, there has been this profound reduction from about 1985 on, when all of us started on this ride, when the people who are my siblings in science, and that's what I'm going to talk about. That red dot, I believe, is where technology and science came together to fundamentally change understanding of disease and then understanding of how to translate science into interventions. Now, the pictures here are two people who were instrumental in that dot. The first is Howard Lord Florey, who won the Nobel Prize for the synthesis of penicillin, but came to visit Harvard Medical School to give the Dunham Lectures in 1964. The person on the bottom was Morris Karnofsky, who's the most quoted author in pathology, written 6 citation classics, discovered the blood-brain barrier, invented immunohistochemistry, and was my postdoc advisor for 6 formal years, but really for a lifetime. And when Florey met Karnofsky, he returned to England to, in 1966, write a paper which was called succinctly The Endothelial Cell. And when he was commenting on the fact that Morris had, in that time, identified the endothelial barriers in the kidney, in the brain, in the thymus, using very sophisticated chemical techniques, he said that he envisioned a time when new techniques are certain to be followed by new discoveries, and the pushing of a known technique to greater heights of technical achievement will produce new accretions of knowledge. So translated from British English to American English, what that means is that there is a moral obligation as scientists to make your work impactful for society. And that the more you understand about the societal problems, the more you'll drive your science, and the more scientific knowledge, the more you will impact quality of life around us. The definition of engineering in the Oxford English Dictionary is the harnessing of the language of mathematics and the disciplines of science to improve the quality of life for all. Now, most people don't know that last statement, but I'm very proud to call myself an engineer because it's this dictum that I want to talk about. So sometimes I'm a little embarrassed that in years before many of you were born, I was actually publishing papers. But in 1989, Mike Klagsbrun, who had discovered growth factors, he coined the term FGF1 and FGF2, which were then called acidic and basic FGF, and he and I wrote a paper predicting that in addition to atherosclerosis, there was a process that would occur, especially when you intervened in an artery, which we now know of as intimal hyperplasia. I was a graduate student with Bob Langer, and I was increasingly interested in cardiology as I was going into that field. And I went to speak to Morris, who is a lovely, lovely man, but a little gruff. And I said, Professor Karnofsky, I'd like to work with you on understanding this process. And he said, Elazer, I need to tell you something. I said, oh, go ahead. He said, the dumbest blood vessel is smarter than the smartest vascular biologist. I said, oh, yeah. He said, no, you gotta let me finish. I said, okay. He said, and the dumbest vascular biologist is smarter than the smartest cardiologist. So what can you bring to our laboratory? He said, I said, well, Professor Karnofsky, I'm an engineer, and we're dumber than everybody, but we can make things work. And he looked, I couldn't tell what he was going to do. He laughed, and he said, okay, you're here for 4 weeks. And so about 20 years later, I actually officiated at his funeral. But the idea was initially that we were going to try to recapitulate this process that you see in the lower right. That big donut is all hyperplasia of cells. And in the upper left are outside the blood vessels, all this neovascularization. Now that actually is a remarkable slide for 2 reasons, because it was the first time anyone had ever stained an artery with 2 antibodies at once, because don't forget, Morse had invented immunohistochemistry. So we were able to show in the upper left and the lower right using double immunohistochemistry, which today seems absurdly simple, but in its day was the first time anyone had shown it, what the source of these cells were. And the idea was that we would take growth factors and we would instill them into the artery and we would create the lesion. And lo and behold, using the techniques that I had learned from Bob Langer, I made these devices that would release fibroblast growth factor 2, place them outside the blood vessel, and nothing happened. And Morris, being Morris, said, well, that's a good result. I said, how is it a good result? I just spent 6 months of my 4 weeks not doing anything. He said, well, look at the endothelium, and you can see in that bottom panel there the extracellular matrix with the endothelial cells. And he said, they look beautiful. I said, yeah, well, but nothing happened. He said, yeah, well, take them off. And so we stripped out using the berry balloons that Grunsig was using, and we damaged the endothelium a little bit, and you can see here that multilobulated polymorphonuclear leukocyte there, where some of the endothelium has been disrupted, and then FGF starts to cause more neovascularization and more proliferation, and when you remove the endothelium entirely, and now you allow for macrophages and platelets, is what you see in the bottom panel, you get this profound effect. Now, it was a pivotal paper. It's been a paper that JCI paper in 1992 has been cited almost 1,000 times because we used it to understand this complex architecture between interventions like balloon angioplasty and vascular biology. And as Morris had said, the smartest cardiologist is nowhere near what the dumbest blood vessel can conceive of. And we proposed a theory which is called the Karnofsky-Edelman theory, where the endothelial cells are bioreactors. They're producing substances that respond to their environment, mechanical stress, flow, shear, noxious stimuli, and they do so in a dynamic way. And you might ask, well, of course, we all know that, but you have to understand the Nobel Prize had just been awarded to Moncada and Van Houte for showing that the endothelium was a barrier, that the endothelium, when it was intact, would prevent compounds from coming in contact with smooth muscle cells. And when the endothelium was lost, the compounds which would vasodilate an artery would vasoconstrict. And so I presented these data at the Blood Vessel Club. There's actually something at the Federation meeting called the Blood Vessel Club. Presented that, as did two other investigators who had similar results with FGF. In that same year that we presented these papers, two other groups, one from the NIH, one from Washington, showed that FGF didn't do anything if the endothelium was intact. And Russell Ross, who is known for many as the father of the response to injury hypothesis for atherosclerosis, was the grand man of pathology, and he was commenting after my thought, and he said, "Young man, you're an idiot." He said, and I quote, "If God had wanted the endothelium to be a bioreactor, he would have put it outside the blood vessel." This is the most moronic idea I've ever heard. We went back to the lab, and Morris said, "That was great." I said, "Morris, can you say a sentence that has moron, idiot, and great in it to me?" I said, "No, now you just have to prove him wrong." So I don't know if I can get this to move forward. Aruna Nathan, I charged her with trying to harvest endothelial cells and put them outside a blood vessel. And in a rat, what we showed was that if you injure the blood vessel, you get that big donut, and the size of the donut relative to the size of the outer donut, when they're equal, is a 1. When it's 1.2 or 1.5, it's triple its size. And if you put some gel outside the blood vessel, it actually makes it a little bit worse. And if you released heparin, which was a compound that we had isolated from endothelial cells, you actually dramatically reduce the intimal hyperplasia. But what Aruna did, which was quite amazing, was she actually did harvest endothelial cells. She actually used what today we call tissue engineering. And in papers in 1995, she actually showed that if you take intact endothelial cells, and we didn't know anything about immunobiology at the time, bovine aortic endothelial cells wrapped around the blood vessel virtually obliterated intimal hyperplasia. And if you put in cells that were deficient in native form of heparin, heparin sulfate proteoglycan, they actually made intimal hyperplasia worse. The terrible irony that Russell Ross did was he died 6 months before we published the paper, and I never forgave him for that, because I wanted to send him this paper. Now, what a series of postdocs and graduate students did was actually examine in detail why this works. And so what we're able to do was to show that you can take endothelial cells in 3-dimensional matrices and grow them to vast number and preserve them for long periods of time and carry them around and place them. And then what we showed is that endothelial cells, which we always think of as cobblestones, are actually not cobblestones. They're like your hand wrapped around a strut. And you can see in the lower left those pseudo-colored green cells, and the purple is the matrix struts of that porous matrix. And you see how, if you take your hand, everyone take their hand and put it on a table, and you see that your thumb, it's painful. Now slide your thumb over the edge of the table, and all that stress, the energy stress in your hand, goes away. And that's what endothelial cells do, and we discovered that. And we discovered and we showed in those middle panels in these double dye exclusion assays that when the cells are in that configuration, they can stay viable green for long periods of time. And we actually showed that they actually have all the functional elements of cells. Now, this is the heart of what we learned from Otto Schmitt. Otto Schmitt did all those things that Professor Bischof told us, but he wrote at the end of his lifetime, in his mid-80s, this wonderful piece called A Lifetime of Connections. And he said, biophysics is not so much a subject matter as it is a point of view. It is an approach of problems of biological science using the theory and technology of the physical science. It's also, he said, a biologist's approach to problems of physical science and engineering, although this aspect has been largely neglected. And for the rest of the talk, next half hour, I'm going to talk about how this conceptual idea drove a lot of our work. This is a common notion of what an endothelial looks like, cobblestones, but using sophisticated imaging, which we actually showed is that endothelial cells are like wheat in a field. They actually are anchored and they can flow, they can move as blood flows over them. And that when you actually are putting endothelial cells into three-dimensional matrices, you're actually recapitulating the very essence of how they have evolved to sit within blood vessels and bone marrow. In fact, Salic and Marie Caputi looked very critically at making a whole variety of matrices of different porosities and different moduli of elasticity, and she showed that the amount of heparan sulfate proteoglycan and the fractional inhibition that endothelial cells in different matrices could elaborate on smooth muscle cells were directly related. And fascinatingly, for the work that goes on here and Brenda Ogle's Stem Cell Institute and others, the modulus of elasticity of extracellular matrices and the point at which endothelial cells on scaffolds are identical. They're somewhere between 1 and 3 kilopascals. We should have known that by first looking what is the modulus of elasticity that supports endothelial cells in nature, but we backed into it in a different way. Now, in the spirit that many of his people have looked at, can't stop at small animal experiments, went on to large animal experiments. We didn't stop at large animal experiments, we went into humans with this. One of the big problems in dialysis and vascular access, whether you take a graft, as you see in the upper left, a synthetic polyethylene graft, or you attach an artery in a vein, is at the heel or toe, you get this profound amount of intimal hyperplasia. There's a rind there, which I think I highlight here in green, where all of this growth occurs. Now, what Ellie Nugent did, and you can see the sponge in those diagrams, is she wrapped some grafts with empty sponges and some with endothelial cells. And the ones with endothelial cells actually had remarkable effects, almost total obliteration of intimal hyperplasia. But in a fascinating way, you can see those inserts, two bottom panels, marked reduction in the inflammatory response. She actually, and we don't have time to go through it, drove this process with one of our industrial collaborators through to human clinical trials. And in a human clinical trial, she actually showed that patients who received just standard AV fistulas would have thrombosis or infection at 2 or 4 weeks at about 35% of the time. And if you place these things, you actually see within them that they generate antibodies to their own endothelial cells 56% of the time. Now, it elaborated a theory that endothelial cells are sided cells. They have a top and a bottom, they have a front and a back. But what was fascinating is we postulated that the reason the matrices work is because the epitopes, the identifying antigens, are on the bottom, and when the cells are anchored, they're not exposed, and when they're pulled up, they are. And so even in patients who have nothing foreign put in, their own vein and their own artery, 56% of them will have autoantibodies. When you put in these matrix-embedded endothelial cells, you not only reduce by 4.5-fold the risk of thrombosis and infection, but you actually reduce the antibody response. Now, we had put bovine aortic endothelial cells into rats and not gotten an immune response. We didn't immunosuppress them. And these are not autologous cells. These are allogeneic cells. These are cells from a cell bank that we had banked from another human. Because they're matrix embedded, they don't generate an immune response. Heiko Mita, who's a cardiologist now in Germany, had actually shown that when you take endothelial cells and you inject them into pigs, you generate this huge spike in IgM in green and IgG at the bottom. If you matrix embed the cells, that almost totally goes away. He further showed that you could actually re-challenge these pigs at 90 and 120 days, and if you use free endothelial cells, they actually get this anamnestic, this hysteretic, this memory response. But if the cells are matrix embedded, that doesn't occur. Now, what I'm excited about is most recently, that is last year, we published a series of papers which I think Otto Schmitt would have liked. And that is, we showed using single-cell sequencing and primary component analysis that cells that are by their nature sided, epithelial cells or endothelial cells, cells that sit on the surface of tubes, unlike cells which are structural bricks of tubes like fibroblasts, have a profoundly different cellular signature, a genetic fingerprint in 2 dimensions and 3 dimensions. And what those violent red panels at the top show is the difference in gene expression in 2D and 3D. And you can see on the far right, the fibroblasts are virtually identical, whereas cells that are layers, cells that sit at luminal interfaces, are very, very different. And you can see on this primary component map, this PCA map, that epithelial cells, endothelial cells from different vascular organs are more like each other than in the various dimensional forms than the difference between the dimensional forms. In other words, in 3D and 2D, there's a profound difference. But what most people call endothelial heterogeneity, differences in endothelial cells between a lymphatic, a venous, and arterial and arteriolar endothelial cells, is actually quite minor. They're all in their individual boxes. But whether they're in 2 or 3 dimensions is not. Now, I want to take us back to the dawn of stenting. This was the placement of the first biomimetic stent, if you will, what became the multi-link stent, which is today the Xience family of stents in pig. We did this with Morris, Campbell, Rogers, and I. Those are stents, 3 stents outlined in white. And what we had shown was, I had asked Campbell, who was then a resident in medicine, I said, we have these devices. They fail 5 to 12% of the time immediately because they clot. 40% of the time they have this intimal hyperplasia. I think we have absolutely no idea what's going on a cellular basis. Let's put them in. And my prediction is that we're going to see that we wipe out the endothelium, because I had just spent the previous 6 years of my 4 weeks with Morris examining the endothelium. And I predict that you're going to wipe off all the endothelium right near the stent struts, being the great scientist that you heard Dr. Bischof say that I am. And Campbell came back, and he had used a preparation which Morris had told us to use, which was then 150 years old, which is silver nitrate staining. And what you see is the beautiful cobblestones of the endothelial cells. In panel C, you can see the cobblestones. I'll explain what those little dots are in a minute. And you see outlined in yellow where the endothelium is denuded, those wavy lines are where the stent sat, and it excluded the silver nitrate. And precisely where I said there would be endothelial denudation, there was endothelial preservation. And precisely where I said there would be endothelial preservation, there was endothelial denudation. What we realized was that I thought the stents were working like windshield wipers. But in point of fact, the balloon was protruding through the stent struts. And so, being clinician investigators, we worked with companies to first make non-compliant balloons, because the initial balloons were very compliant, and then to make stents where you would minimize the area of protrusion and maximize flexibility. Now, at the time, Dr. Kuntz will tell you, this was almost heretical, because contrary to Morris's dictum about the dumbest blood vessel, the smartest cardiologist, industry believed that a stent is a stent is a stent, and you couldn't make a better stent. They were so adamant about that most companies didn't have development programs, and none of them thought of ever patenting anything beyond that. So in this paper, this pivotal paper in 1995, we actually made 2 different stents. Made a stent, which was that finger handcuff-like stent you see above the yellow bar, and it's in that electron micrograph in its unexpanded and expanded state. And then we made a stent that we had used in 1991 from 51 CAD/CAM drawings to maximize flexibility and minimize balloon protrusion. And we showed that every element of the vascular response, thrombosis, inflammation, in particular of macrophages, and intimal hyperplasia, was reduced not simply, by creating a better device. It really does adhere to this notion of trying to harness science to drive technology. And in particular, we did 2 things. On the bottom panels was the first time anyone had ever shown the histopathology of stents. It's actually really hard to do, because histopathology at that point required that the embedding media be harder than material around it. So you can't use glass knives. We had to use metal knives. And I used to send Campbell to the naval shipyards, which were 190 miles south of Boston, every Saturday, where we would pay the shipyard metallurgist to cut our stents. And then we actually worked with Leica to invent a microtome which used diamond knives. So an offshoot of this work was, first, we invented the first microtomes that could cut metal on 5-micron scales. And then second, we showed immunohistochemically something which took ages to publish, but that the lesion was not comprised of smooth muscle cells, but of inflammatory cells. Now, The stents had reduced the failure rates of angioplasty from 40% to 25%. But the really remarkable thing was that 3 months or a few months after the introduction of that paper, a clinical trial, 2 years after the introduction of that paper, the West 1 trial was formed, and it actually did as well as bypass grafting in terms of its failure rate. In a very short period of time, and this was what's so exciting about all of us who were in the room at the time, we had actually taken a technology which failed as often as it succeeded into one which was as good as the most effective therapy. Now, there was still a nagging lack of understanding of what was going on with the endothelial cells. Here's, in that panel A, you see the white curve, which is where the stent strut was. You see the cobblestones. When you balloon denude an artery, you see all the basement membrane align. If you wait 3 days, the endothelium is restored. Now, I'm often amused when I read papers that talk about endothelial restoration because those endothelial cells don't look healthy. They look cuboidal. They don't look spindle-like. They are very heterogeneous. They don't form gap junctions. And yet, even today, people talk about, is the endothelium restored? And so what we beg people to do is think differently about endothelial cells and endothelium. David Ettinson, in a really important paper, showed that if you harvest cells from these different sites, the ones that are even from the site where they had been immediately denuded are profoundly anti-proliferative and anti-inflammatory, but the ones where the endothelial cells are restored actually promote inflammation, thrombosis, and intimal hyperplasia. So most recently, Adichie Kalluri actually looked at this on a genetic fingerprint level. And this is the wonderful thing about existing in the environments that we live in. We're not bounded by technology. We have a full range of technologies available to us. We can use mechanics and fluid dynamics, but we can use single-cell sequencing and omics and transcriptomics and proteomics and metabolomics. And in this study, we harvested cells from aortas, and we showed for the first time that vascular cells could be identified by specific marker genes, fibroblasts like we had talked about, vascular smooth muscle cells, monocytes, macrophages, red blood cells, and endothelial cells. And they all form different clusters on these Tisney plots. And in an unbiased way, machine learning can actually extract and identify what the cells are by the 5 genes of highest differential. And we actually showed, in a way now that begins to explain what is going on when you intervene on this heat map, is that the cells that are the primary bricks and mortar of a blood vessel are fairly monolithic. But the cells that are residing at luminal interfaces undergo a very profoundly heterogeneous effect. So endothelial cells exist in 3 different forms. Monocytes and macrophages exist in 3 different forms. Fibroblasts and vascular smooth muscle cells are always singular. And what we're able to do was to define these forms and to actually ascribe to them very specific, and I'm going fast through this on purpose because the specifics are relatively unimportant, but we could identify with endothelial cells in a specific state a specific function. One form of endothelial cell is brilliant at extracellular matrix organization and integrin signaling structural. Another form is really important for lipoprotein handling and another form for neovascularization. In stark contrast, vascular smooth muscle cells are always monolithic. And it doesn't matter what you do to these cells. You can put animals on high-fat Western diets or low-fat diets, and yet we still see this same clustering. Now, we often take the lab on outings. One of the outings was whitewater rafting. In that raft, you can't see it to now, there were 3 rafts that day. That raft has the faculty of most of the biomedical engineering departments in the United States. They're all frightened. You can't see their faces. But when we came away from that, Kumaran Develu and Brinda Balakrishnan had the idea that we really ought to be examining the fluid dynamics, because I became Morris. You become your father. I said, guys, this is brilliant. There's white water in the raft, and they're all saying, why did we do this? We wanted to go bowling. And what we began to look at was, can you look at the recirculation zones around stent struts, just like you can look at the recirculation zone around your hat, around the rocks in whitewater. If you ever fall out of a whitewater or your hat falls out, you look right before or right after the stone, because that's where the water is stable. And this paper has become a citation classic because it actually showed for the first time why thin strut stents are better than thick strut stents, and actually graded this concept of malapposition. How does a stent strut, which should be adherent to the wall in that bottom red-green, as opposed to the top where the struts, inflexible, are hanging out in the breeze? And that panel, that series of panels on the left shows that the flow domains are very, very different depending if the strut is near the wall or in the field. And what it did in that time was generate a whole collaboration with a variety of industry of how to make better stent struts. In a paper that'll be published later this year, we actually showed that you can begin now to use single-cell sequencing using UMAP, not just TISNE, and stochastic processes to differentiate cells on the basis of low altered shear stress or high physiologic shear stress. And the blue and the red cells show this clustering in a way which is profoundly different. What's even more important is that for the first time, we're able to understand this great debate in vascular biology and fluid and hemodynamics, that we rarely have a Reynolds number high enough to have random flow. There really isn't turbulence in blood vessels. It almost never occurs. We have flow separation, but we understand that what turbulence means is yellow. Turbulence is that you can define both normal or physiologic blue and altered recirculation in red, and the turbulence is where you have both flow domains. And I'm not going to go through this, but it's an extraordinarily exciting finding because we have on a genetic level for the very first time proved something that we only understood at a level of fluid dynamics. Today, there is a rule as to how you make stents to minimize the flow disruption, the balloon protrusion, and maximize endothelial preservation. And every company that makes stents uses the work of these two people who are now professors in their own right. And every engineer at a stent company knows this work and uses these rules, and every regulatory body insists on these kinds of maps in looking at endothelial, at stent biology. Now, what we can do today using generative AI is actually predict in advance not only what's going to happen from the design, but what's going to happen when some of the living heart images, for example, that Paul showed me this morning, can actually intervene. And so we can do a virtual clinical trial now using generative AI, using OCT images. We can characterize and classify and actually do virtual interventions as part of this to actually understand how to maximize the implantation of these devices and use this on the basis of, let me see if I can make this work. We can now correlate an OCT image from a patient. We can now infer what an OCT will look like from a digital twin of that patient. We can actually recreate an artery to look anything we want. We can use any stent design we like. We can put it in, and we can actually measure or infer what the genetic response will be. Ron Siegel and I, in 1979, joined the laboratory of a guy who was destined to be nothing, according to the graduate officer of the Department of Electrical Engineering and Computer Science. That's Bob Langer. What Bob asked Ron and I to do was to take this, which was an engineer's view of an innocuous portable drug delivery device. According to an engineer, it's an engineer. I always joke, my son said, it's an engineer because of the white shirt and the pocket protector. But that purple canister is drug, and that image that my children make fun of is one of a few images that Ron sent me that we had. On my fingertip is a drug delivery device that for the first time in 1979, 1980, we were able to encapsulate, in this particular case, enough insulin to keep a diabetic animal euglycemic for 40 days. That's a ping pong ball. I always joke they worked with Bob Langer before he was Bob. I had to actually steal that ping pong ball because we didn't have enough money to buy ping pong balls. Now, what we did was actually Xiaowei Wang and David Wu, who are professors in their own right now, I asked them to take that little pellet and put it on a stent. This is the image of the first drug-eluting stent. Looks beautiful, right? It's like someone dipped metal cage into cotton candy. And there's a fluorescinated drug. And that image is now a classic image in medicine because it's a confocal image 200 microns deep into a blood vessel. And what it shows is 200 microns deep into the blood vessel, you can still see the imprint of the stent. And it proves unequivocally that drug elution from stents could work. Now, this is why I say I'm tremendously proud of our work with industry, because that actually is the same stent coated with a 2 to 5 micron coating by our industrial collaborators. If we had had to use our cotton candy stent, for sure, it would have been a disaster. So in my lifetime, we went from that backpack to a pellet to cotton candy to a device which is now placed in millions of people each year. The first paper that described this work actually was by Doug Drachman, Campbell Rogers in our lab in 2000, and it shows how the intimal hyperplasia is virtually obliterated by actually eluting the drugs from the stent. At the time that we were putting stents in, 43% of that middle gray bar, metal stents would have enough intimal hyperplasia, enough restenosis to have another procedure. The remarkable thing was, and there have been very few things in medicine that have worked as profoundly as drug-eluting stents. maybe antibiotics, you almost entirely obliterate that response. Now, there was a little warning in that paper, and that is if you give too much drug too fast, you actually can create thrombosis. And that's something we identified in 2000, and to this day remains an Achilles heel of the work. But within 10 years of that first angioplasty study that I cited, the drug-eluting stents actually surpassed the survivability of bypass surgery. Now, to be fair, what's happened is only the worst, only the most complicated, only the most difficult go to bypass surgery. Everything else goes to stenting. I'm going to skip over this for the sake of time. I am going to tell you something interesting, though, using single-cell sequencing analysis. And that is that there are two drugs that we use principally for drug-eluting stents, a drug called paclitaxel and a drug called sirolimus. In a paper that will be published later this year, what we actually showed is that you, for the first time, can understand that there's a commonality and a difference between these drugs. It is always the case that when you give the drugs and you alter flow, you can distinguish the altered flow response. But the drugs actually act profoundly differently. So the question is, how do they work in a similar fashion? So you can see on the left, a drug called paclitaxel, on the right, this drug rapamycin or sirolimus. You can see within each panel the overlay of, in the control cells and the drug cells. They're always in one giant oval, separated by a bridge to the same drug and control cells when they're altered flow. But what I'm going to skip over right now and make this really important point is that the commonality of these things is that, if you remember, I said that I failed to have FGF do anything when the endothelium is intact. When flow is normal, endothelial cells have on the dendrogram 1 and 3 many, many branches. But if you use altered flow, they have many fewer branches. What does this mean? This means that endothelial cells are, by their definition, a cell which has no state. It has no single stable state. You can never identify its phenotype. It is necessarily a cell which always responds to what's going on around it, and in particular flow. And as long as the flow is physiologic, it can generate a genetic response to drugs and mechanical stress and other things. But as soon as you alter flow, you transform the endothelial cell into a state of limited phenotype. It's a profoundly important finding because it begins to explain why it's important to minimize the injury, because you need to preserve the viability and integrity and flexibility of the endothelial cells. And if you start out by injuring it, the ability to use the drugs to have a genetic alteration is substantially reduced. I'm going to skip this again for the sake of time because I want to end at 1. I want to end with what we've learned beyond stents, because adults are not the only people in the world. Atherosclerosis is not the only disease in the world. And so we use a lot of these lessons recently to do something I'm really proud of. We took everything we had learned from the drug-eluting stents and we began to look at diseases which were a real problem. One of the most common congenital diseases is heart disease. And it's a lifetime burden for children and their parents. It requires multiple operations. Some 6,300 children are born each year with pulmonic valve disease. There are over 130,000 patients living in the United States with these diseases and 10 times as many around the world. When children are born with these congenital diseases, they usually undergo a modest corrective procedure when they're young, but it oftentimes leads to this profound dilation of their ventricles. It's not a definitive, it's palliative surgery. And then they have further delayed definitive surgery as they undergo these pretty harsh medical managements and very restrictive lifestyle until they can actually get the definitive surgery. One of the reasons that surgery is delayed is that from the age of 2 to 16, you see in the upper right, the diameter of the pulmonic valve grows substantially. It doubles, diameter doubles. And yet, the valves that we have are of a fixed diameter. The problem is that if you implant a valve in a child after they're 10 or before they're 10, you get a very different response to the placement of the valve. In fact, more recent studies have even looked at this. When you put a valve in at a child that a person with congenital heart disease after they're 40, they survive without free of events for a very long time, 80% of the time. But if you put the valve in when they're less than 1 year old, that actually drops off tremendously. So most clinicians delay pulmonary valve replacement until children reach near adult size. And the question we asked was, could we take all the learnings from stents and now apply it to valves? Well, in the spirit of biomimetics, we started concentrating not on arterial valves, but on venous valves. Our veins have 7 times the capacity of blood of our arteries. And that's because they have a very different aspect ratio. So your veins have valves that when they're depleted, a very, very high aspect ratio, and when they're full of blood, a very low aspect ratio. And so we wondered, could we make a valve that actually operated in the same way? And so Sophie Hofferberth, who is a very talented surgical resident, and Pedro Del Nato, who is the chair of pediatric surgery at Children's Hospital, and I created a valve that used many of the structural elements of stents that could expand or be expanded in the very same way as venous valves work, retaining coaptation at the same time as allowing them to be implanted at very young children. This is truly following Schmitt's dictum of biomimetics. What I want to share with you in closing is that in a very short period of time, we filed the patents, we got an NIH STTR phase 1 grant, long-term animal grant. We've now implanted this device in 10 children. We've completed the early feasibility study, and we're moving to a definitive study in the shortest period of time. And the reason that that worked was because we had used all the science that we had learned from one domain to carry something into another domain. We had done what every engineer should do. We had addressed a critical unmet need by using biomimetic analysis, leveraging all that we had done, and then driving it into people. And what I'm going to end with is to say that it's really an honor to be here, honor to give the Schmitt Lecture, an honor to be here in front of longtime friends, people I've admired to this extraordinary institution, because everything that I've been associated with adheres to this point of view that Dr. Schmitt elaborated about biomimetics and biophysics. And I'm deeply indebted not only to Professor Karnofsky and Langer, but to what is now about 370 postdocs, fellows, and graduate students who have shouldered this burden. And what I would, and of course to my family, who let me have the most wonderful job in the world. None of my children are physicians. But I would end by saying that we therefore have an obligation. René Laennec invented the stethoscope in 1813. Nobody believed him. No more people didn't want to believe that technology could help medicine. And so in 1816, he wrote this incredible tome called La Auscultation, Mediate Auscultation, putting something between your ear and the patient's chest. And here he is surrounded by his students and the nurses and junior physicians at the hospital. This is me, 200 years later. And I'm using an electronic version. I'm using generative AI to display the signals. But I'm essentially doing the same thing that Laennec did. There's a paper we published in the New England Journal of Medicine about what our responsibilities are to seize on the legacy of history of innovation, but above all, to teach the next generation of, like all of you in this room, about being a conceptual revolutionary. So thank you very much for this opportunity to speak. Happy to entertain any questions. Stay around. I know it's one Sure, I'm not going anywhere. And I'm going to blow up the question a little bigger. So oftentimes we speak about all the things that worked, but we don't talk about things that didn't work. And you learn more from the things that didn't work than the things that did. So radiation, extremes of temperature, infusing many harsh drugs, all worked really well in animals, and they were a disaster in humans. And that's why I think that the single-cell sequencing papers that we've written in the last 2 years are so vitally important, because they help us understand the blood vessel is not a plastic tube or a metal tube. It's a living thing. And what you're trying to do is preserve the physiologic architecture and function of the blood vessel. And if we've learned anything from the last 30 years of intervention, is that the harsher the immediate injury, the worse your late-term response will be. And so what cryoplasty did was secretize or scar the artery, and you would have a profound compliance mismatch with virtual cross-linking of collagen, obliteration of elastin, and lack of any meaningful, viable cell recovery or transport across. And so one of my pleas has been from the beginning is that while Rick Kuntz was developing all these statistical methods for clinical trials, is to use the same rigor for preclinical work, but not to call it preclinical. Because the implication is that the obligation to do the science stops once you enter the clinic. And that's when I think it begins, and it's in particular with the failure modes that we need to do the most science.
Dr. Edelman, wonderful talk. You were referring to the dynamic nature of endothelial cell interactions. And if we consider endothelial response, it happens in seconds, interacting with the neurons, endothelial cells, pericytes, and astrocytes. But when we're investigating changes in endothelial response and endothelial function under pathological conditions, we rely on more static measurements like changes in gene expression, changes in protein expression, rather than looking at changes in the dynamic interactions. So do you think that we are missing the point there?
So my PhD is actually in physics, even though it's in electrical engineering and computer science, in a field called continuum dynamics, which sees the world as a continuum, both a temporal continuum and a spatial continuum. And so what we did here was to take single-cell sequencing and actually plot trajectories. And what this means is it's not just a cluster of cells, is you can actually track the cell as through time it actually chooses where to home in on its trajectory. And what you can see is that the physiologic response in those whisker plots on the bottom show these broad boxes, the blue and the orange. And they retain, endothelial cells retain the ability under physiologic flow of dynamically sampling everything around them. But as soon as you lock the flow, you lock the endothelium. You remove its fundamental existential definition of a cell that is always dynamically sampling and responsive to locking its state. And so what you said is perfect. And it's this kind of analysis, which is actually really hard to understand.